Namaste friends, welcome back to Banu Biology Center for True Learning. In this video, we will talk of Mendel's principles or laws of inheritance. Based on his monohybrid experiment and dihybrid experiment, so he proposed totally 4 principles. Let me list out them one by one. The first principle laid down by the Mendel is principle of paired factor. Principle of paired factor. The second principle is principle of dominance. Principle of dominance. The third principle is principle of segregation. Principle of segregation or principle of purity of gametes. The fourth principle is principle of independent assortment or principle of free recombination. These are the 4 principles given by the Mendel from his experiments. Out of 4, only 3 principles were get the position of the laws. The status of law for these 3 principles was given by Carl Correns. There was one scientist, Carl Correns. Carl Correns was a scientist who gave the status law to these principles. To this one, the principle of dominance, and also for principle of segregation, and also for principle of independent assortment. Now we can refer these as law of dominance, then law of segregation, then law of independent assortment, or law of free recombination. So let us learn each principle. So what this principle says, how this principle was actually laid down by the Mendel. First one, principle of paired factors. As we know come to know the factor given by the Mendel that whatever the unit factors or whatever the discrete unit factors responsible for a particular character always present in a pair. So what the principle of paired factors says to us? The unit factors always occur in a pair. For example, the genes responsible for tall plant capital T. These two genes always present in a pair. Another example, the genes responsible for dwarf plant or short plant always appears in a pair, that is capital T and another in capital T. So this is the very simple the principle. Next come to the second principle or law, that is law of dominance. So as the name indicates, something is going to be dominate the other. If you are able to remember the monohybrid cross, especially the F1 generation in monohybrid cross. So it is like this. So let me give an example. Let me explain this with an example. This is the parent generation in the monohybrid cross. One plant is taken as homozygous dominant, that is capital T capital T. The another parent was taken as homozygous recessive plant, that is small t small t. The gametes produced by these plants, always all of them contain capital T allele or gene. The gametes produced by this recessive plant has a small t allele, right? So in case of this, both are the dominant. There is no dominant, there is no recessive, for example. So now we do the cross. So here we do the cross pollination. So results will be like this. This gamete fuse with this one, capital T small t. Then this plant can fuse with this gamete. So these are the probabilities I am telling you, right? So any gamete can fuse with any other gamete. So here I am writing you the probabilities. Then come to second gamete. This gamete can fuse with the first gamete, that is capital T, then small t. So again this gamete have chance to fuse with this, then gamete becomes capital T small t. This generation referred as first filial generation, F1 generation. If we see the phenotype of these plants, all the plants are produced in the F1 generation are having the same genotype and also express the same phenotype. So let me write the phenotype of these plants. So all these plants express the phenotype of tallness, that is heterozygous tall. So what happened here? So in case of parents, both the alleles are same. So either both the alleles are dominant, so either both the alleles are recessive. But when we come to the F1 generation progenitors or offsprings, there is presence of two dissimilar gametes. So one is genes, one is capital T, another one is small t. So even though two dissimilar genes or alleles present, all the plants became tallness. Here we need to do an important observation. So what happened here? So only one allele, that is capital T, right? This capital T allele act as dominant allele, then this small t allele act as recessive allele. So in case of this plant, so one gene or one factor suppress or mask the expression of other allele. This is what the law of dominance. What is law of dominance? One factor or gene mask the expression of other allele is called law of dominant. So in this simple example, the capital T allele suppress the expression of the small t allele. Here the capital T is going to dominate the small t. So this is what the principle of or law of the dominance. Next come to the another law, that is the law of segregation or purity of gametes. Here the segregation is nothing but separation. Okay, na? The meaning of segregation is separation. So what is going to separate? So during the gamete formation, alleles occurs in a pair, separate or segregate and enters the different gametes. That is called the law of segregation. So I explained this law so with the same example so I gave here earlier, right? So when we wrote the gametes, so this is the parent plant has two two alleles, capital T, one is capital T, another one is the capital T. So during the meiosis, right? During the meiosis, these two gametes separate, right? This capital T enters this gamete, so this capital T enters this gamete, right? Right. So once again, so we take this example. For suppose the plant has capital T and small t alleles, so it is going to form the gametes or produce the gamete. So it produces two types of gametes. So one type of gametes contain this capital T allele only. So other gametes contain this small t allele only. So during here the meiosis cell division takes place. So one important point I would like to tell here. So at the time of Mendel there was no concept of meiosis, right? So later on the meiosis concept was discovered by the other scientists. So anyhow, so during the meiosis, factors, unit factors or discrete unit factors occurs in a pair, the separate and enters the different gametes. So here we can ensure the purity of gametes because only one allele is present in a gamete. So this law of segregation is also called a law of purity of gametes. There is no contamination of the gametes. So it is called purity of gametes. Next come to last principle, that is the principle of independent assortment or law of independent assortment. So before that, so I would like to tell so how these three principles were laid down. Based on what? So based on which kind of observations? The first three principles are given by the Mendel. So these principles were completely based on the observations made from the monohybrid cross. Monohybrid cross. So after successful completion of the monohybrid cross, so Mendel did some observation. Based on those observations, the principle of paired factors, the law of dominance and the law of segregation or purity of gametes were laid down very clearly. So monohybrid cross results are the basis for development of these principles. Next, come to the last principle, the law of independent assortment. So what is law of independent assortment? So during the gamete formation, a pair of factors or pair of genes responsible for a particular character independently assort and enters the different gametes. So to understand this principle, so we need to remember the F2 generation of dihybrid cross. Actually this law was laid down based on the results of dihybrid cross. So let me briefly explain the F2 generation of dihybrid cross here. Dihybrid cross. So directly, so I write the combinations, four type of combinations so we received in the F2 generation. The first one is round seeded, round shape yellow color seeds. Then second combination round shape, then green color seeds. Next third combination wrinkled shape yellow color seeds. The last combination wrinkled shape green color seeds. If we give an observation here, these two are the parental types. They are like parents, but these two are the new combinations. So this is the proof enough to say there is the independent assortment of a pair of factors during the gamete formation. So If it is not possible, if there is no independent assortment, so there is no the freely separation of the pair of genes and enters the gamete, so there is no chance for getting these two new combinations. One is round green, another one is a wrinkled yellow plants. Wrinkled yellow plants. Of course, there are some exceptions. So after the Mendel, so many other experiments did by other biologists also. Finally, they came up with some exceptions for the Mendel's laws. So let me write exceptions. There are two exceptions for the principle of dominance. Principle of dominance. Exceptions for principles of dominance. The first one is codominance. The another one is incomplete dominance. Codominance and incomplete dominance. In case of codominance, so both the alleles, both the factors equally influence the character. So no gene is dominant, so no gene is the recessive. Both are equal. So it is completely deviate from this laws, right? The second one is incomplete dominance. Here the dominant allele cannot suppress or mask the influence of the other allele. So it is named as incomplete dominance. So we will talk of these exceptions in detail in further videos. Next, the principle of segregation. So it is one and only law, so which is accepted as universal. This is the universal law of inheritance. The law of segregation or law of purity of gametes is the universal law of inheritance. Finally, the principle of independent assortment also has one exception. That exception is linkage. So actually, the Mendel did not consider the concept linkage during his experiments. So after that, T.H. Morgan came up with this concept and said that so this principle was deviated from this, the linkage. So we will talk of the concept linkage also in the further videos. This is about the Mendel's principles or laws of inheritance. Thank you for listening this video.